Hello everyone. So today the topic of discussion will be amino acids and proteins. So I will be taking up the topic in a small parts. So let me share the screen first. Okay. So the topics is amino acids and proteins. Talking about the proteins, they are linear, unbranched, polymer constructed from 20 standard alpha amino acids. And these as amino acids are encoded in the DNA of the genome. So amino acids, if we draw the structure of the amino acid, it is something like this. So this is a general structure of amino acid. So amino acids, they are composed of a amine group, a carboxyl group, a hydrogen atom, and a distinct side chain, and all they are bonded to a carbon atom, that is the alpha carbon, right? So this structure is common to all, but one of the alpha amino acid, that is a proline, is a cyclic amino acid, and this is the exception. So R group or the side chains attached to the alpha carbon is different in each amino, in each of the amino acid. In the simplest case, like if we talk about the structure of the glycine, it is like this. It's a simple hydrogen amine group, right? So if I talk about the lysine, another amino acid, the structure is NH3 plus CH2, 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 CH, COO minus, and the amino group. विनीत तुम अंदर चले जाओ ना, तुम इतनी खांसी है तो ये तुम्हारी खांसी बीच बीच में आ रही है, रिकॉर्ड ही तो चल रहा है. So if we count, it is 1, 2, 3, 4, 5, 6. This is alpha, this is beta, gamma, delta, and epsilon. Right? So in alpha amino acids, both amine group and the carboxyl groups are attached to the same carbon atom, this carbon atom. However, like many naturally occurring amino acids, not found in the proteins, have the structure that differ from the alpha amino acid. So in these compounds, the amino acid is attached to a carbon atom other than the alpha carbon atom, and they are called as beta, gamma, or any other accordingly, right? So this is a small briefing about the amino acid that it contains. Quickly we will revise it. So it contains a carbon which is attached, which is also called as an alpha carbon atom. This is attached to a amino group. Then we have a carboxyl group. Then we have a hydrogen atom, and then we have a side chain. This one. Okay. Now, like there are certain properties which we will be discussing about the amino acid is okay. Now we will be discussing about the amino acids. They can act as acids and base. Okay. How? When the amino acids they are dissolved in water, so they will be existing when an amino acid is dissolved in water. It exists in solution as a dipolar ion, which is also called as zwitterions. So this zwitterions can act either as acid, that is the protein donor, proton donor, or a base where this is a proton acceptor. That is why the amino acids are also called as amphoteric molecule. So at high concentration of hydrogen ions, it means at low pH, the carboxyl group accepts a proton and becomes uncharged, right? So overall charge on the molecule is positive, right? So we can say at low pH, the carboxyl group, they accept protons and becomes uncharged. So overall charge on the molecule is positive, right? So at high pH, if I'm talking about high pH, means there is low concentration of hydrogen ions. The amino acids, it loses its proton and becomes uncharged. Thus, the overall charge on the molecule is negative, right? So this is how it acts like a acid and a base. So I'll just show you a equation. Like we have okay, so this is the condition at the intermediate pH. That is when yes. So at high pH, what will happen? The amino acid will loses its proton and it will become uncharged. Okay, and the total overall charge on the molecule will be negative. And if it is at a low pH, then the amino acid will accept a proton and it becomes uncharged, so that the overall charge on the molecule is positive. So this is at the low pH and this is at the high pH. Okay. Okay. The next one is the optical properties of the amino acid. Let us discuss about this also. Optical properties. So all the amino acids except the glycine, this is except this glycine, all are optically active. If I'm talking about optically active, means that they rotate the plane of plane polarized light. And optically active molecules, they have an asymmetry such that they are not superimposed on the mirror image. So this is the characteristic of a substance that contains a chiral center. A chiral center means a tetrahedral carbon atom with 4 distinct constituents. It is called as a chiral. So the one amino acid not exhibiting a chirality is glycine. Since R group is a hydrogen atom, that is why the glycine is exceptional. So, and one thing more, the chirality describes the hardness of a molecule that is observable by the ability of a molecule to rotate the plane of polarized light either to right, that is a dextrorotatory, or to the left, that is a levorotatory. And we all know that the optical activities, they are measured by polarimeter. So the magnitude of optical activity is measured as the angle of rotation. So angle of rotation will tell you about the magnitude of optically Optical activity. So the quantitative measure of optical activity of a molecule is known as optical rotation, and it is denoted by alpha. Optical rotation of a optically active compound, it depends upon certain factors like the concentration of the compound, light path, length, wavelength of the polarized light, and the temperature. So these are certain factors on which the this optical rotation, or you can say the optically active compound, depends upon. Okay. Okay, next topic which I am taking right now is absolute configuration. So amino acids with chiral center gives two possible isomers, and these are called as enantiomers. So enantiomers, these are the mirror images of each other. So two enantiomers have different configuration. For example, glyceraldehydes. Dextroglyceraldehyde. So these are two isomers. They are called as enantiomers. Uh, See, in glyceraldehyde, they have two enantiomers, and this describe exact the spatial orientation of all the four specifications. So this is called as the absolute configuration. So we can see D and L, they are refers to the absolute configuration of the glyceraldehyde, this one. I'll give one more example of these enantiomers. Uh, Alanine, this is L, alanine, and then we have dextroalanine. So these are two enantiomers. So all the amino acid except the glycine exist in two different enantiomeric form. However, all of the amino acids in protein, they exhibit the same absolute steric configuration, that is the L. Okay? If we talk about in proteins, they will like L-glyceraldehyde. So D form of the amino acids are not found in the proteins. So D forms are not found in proteins, although they exist in the nature. So D form of the amino acids are found in some peptide antibiotics and peptidoglycan wall of the bacteria. So this is one more thing which one has to remember, that usually the D form of the amino acid are not found in the protein. They all have L alpha amino acids. So D forms, they exist in nature and in some peptide antibiotics they exist, and also in the peptidoglycan of the cell wall of bacteria. This is one way of showing absolute configuration. There is another way of denoting absolute configuration using the symbol R and S. Let us discuss that also. So we have R, which is taken, which is from the rectus in Latin, which is called as right, and S, which has been taken from sinister in Latin, which is called as left. So they are used. So here, in this approach, substituents on an asymmetric carbon are prioritized by decreasing atomic number. So we have to see they should have the decreasing atomic number. So configuration is assigned by looking down the bond to the lowest priority substituents and assigning R to the configuration where the remaining substituents are arranged clockwise in decreasing priority. So, and S is then assigned to the molecular form where the substituents are arranged counterclockwise. Like I'll just show you. So this is like R for the glyceraldehyde. And if I want to show L, S. So this will be L glycer aldehyde. So in R and S notation, change of single substituent can change the whole assignment. However, all of the amino acids used in the protein except glycine, which is not optically active, are of the L configuration. In RS notation, cysteine is 2R, whereas others are 2S. This is exception. So, thoda sa, we will be talking about amino acids. So, more than 300 amino acids are present in a cell. Around, there are more than 300 amino acids are present in the cell, but 20 amino acids. They are present in proteins, and these 20 amino acids are called as standard amino acids. We have 21st amino acid also, serinocysteine, and 22nd also, which is only present in RP. So we have 20 L alpha amino acids, and they are specified by simple 3-letter codon. So we have, just to make a list of the amino acid with non-polar side chains, we have 9. We have amino acids with non-polar side chains. We have, there are 9 in number: glycine, alanine, valine, leucine, isoleucine, proline, methionine. Then we have phenylalanine, tryptophan. So if we talk about the PI value, the maximum is for the proline. It is 6.48. I'm talking about the PI value. 6.48, and minimum for the phenylalanine. This is the minimum one with 5.48. We have amino acids with uncharged polar side chain: serine, threonine, cysteine, asparagine, glutamine, tyrosine. Then we have amino acid. They are 6 in number. Then we have amino acid with charged polar chains. They are 5 in number: aspartate, glutamate, lysine, arginine, histidine. So you can see we have 9 polar amino acids. The structure is given. It is showing you the structure. Glycine, alanine, proline, valine, phenylalanine, tryptophan, leucine, isoleucine, methionine. Then we have neutral polar, 6 polar amino acids, serine, threonine, cysteine, asparagine, glutamine, tyrosine. Then we have 2 acidic amino acids, aspartate and glutamate. We have 3 basic amino acids, arginine, lysine, and histidine. Okay. So how can we learn the classification? We can say that the classification of amino acids is based on its chemical structure. If the characteristic, you can say the chemical nature of the R group, we have to see the chemical nature of the R group. Example, if it is aliphatic, then we have glycine, alanine, valine, leucine. If I'm talking about aromatic, then I'm talking about phenylalanine, tyrosine, tryptophan. If it has a hydroxyl group, serine, threonine, likewise. Carboxylic group, aspartate, glutamate. Okay. Okay. Then we have non-standard amino acids also. More than 300 amino acids have been found in the cells, as I have already told you. Besides their role in the proteins, amino acids are and their derivatives have many other biological important function. For example, ornithine and citrulline, they are the key intermediate in the biosynthesis of arginine and in urea cycle. Right? So we know that there are more than 300 amino acids, and some are the derivatives also, and they are playing important role. Like already discussed, ornithine and citrulline, they are the key intermediates in the biosynthesis of arginine and in urea cycle. So in addition to 20 standard amino acids, proteins may contain some uncommon amino acids residues that are formed by the modification of the standard amino acid residues, which are incorporated into polypeptides, like 4-hydroxyproline, which is a derivative of a proline, 5-hydroxylysine, which is a derivative of a lysine, right? So these are certain uncommon amino acids. So it was originally thought that all the unconventional amino acids were made by modification of one of the standard amino acid after it has been incorporated into protein, and a process called as post-translational modification. But amino acids, like there are certain amino acids like selenocysteine, pyrolysine, they are inserted into proteins by translational machinery right? So selenocysteine is introduced during the protein synthesis rather than created through a post-synthetic modification. So it contains a selenium rather than a sulfur of its structural analog cysteine. So since selenocysteine is incorporated into polypeptide during the translation, it is referred to as a 21st amino acid. However, it is incorporated into polypeptide during the translation. That is why it is called as a 21st amino acids. Okay. So I'll stop over here. In the next, we will be doing some other properties of the amino acids. Thank you so much.